Okay, so today we're going to be looking at ways of studying the brain, also known as neuroimaging techniques. So what you're going to be doing today is to understand those different neuroimaging techniques as well as being able to evaluate them. And the four techniques you need to know about are functional magnetic resonance imaging, or fMRI, electroencephalograms, or EEG, event-related potentials, ERP, and post-mortem examinations. But before we go into the way that those different techniques work, you need to understand some key terms, and those are spatial resolution and temporal resolution. Spatial resolution just refers to how specifically a technique can tell us which part of the brain is being active. So incredibly low spatial resolution might be something like it can only tell us which lobe is active, whereas incredibly high spatial resolution will be down to the individual neuron. Temporal resolution is just how specifically a technique can tell us when that part of the brain became active. So very, very high temporal resolution would be able to tell us down to the millisecond, whereas very low temporal resolution might only be able to tell us down to within about 10 seconds or so. So just remember that higher temporal or spatial resolution means that we're being more specific. Okay, so fMRI works by measuring levels of oxygenated blood in different parts of the brain. And the assumption here is that more oxygenated blood going to that brain area means that more energy is being used by that brain area. Therefore, we can assume that more activity is taking part in that part of the brain. So this over here is an fMRI scanner, or an MRI scanner actually, they use the same technology. So this person would lay down here and then it would slide them into the machine, and the output would look something like this. So this is a slice of the brain, and you can see this part here is where the most activity is happening. So fMRI works by blasting them with an incredibly powerful magnetic field. This doesn't do any harm to you, but what it does do is cause the protons of the hydrogen atoms in the blood to align and all face in the exact same direction along a particular axis. What then happens is once they're all aligned, the atoms are blasted with radio waves, which causes them to spin off of that axis. When they turn off the radio wave, the atoms then line back up with the magnetic field. But as they do, they give off an electromagnetic signal, which is what is detected by the MRI scanner. Now the functional part comes from the fact that oxygenated blood gives off a much stronger signal than deoxygenated blood does. And because of that, the MRI scanner is able to work out which parts of the brain have more oxygenated blood, and therefore we can tell which parts of the brain might be more active. I understand that that's quite a complex way to understand how this works, so what I'm going to do is link a video into the Google Classrooms page so that if you're not quite sure of how that works, you can watch that video and it explains it a bit more clearly because it can use more dynamic visuals to illustrate what's happening. And there it is. So some possible applications would be that participants would be given like a wide range of tasks. You can do anything really in an fMRI scanner, and what you would do is watch which parts of the brain become more active while they're doing that task. So just some examples would be that participants might be shown different faces and asked to identify which emotion was being showed, and from that we might be able to work out which parts of the brain are associated with understanding memory, understanding emotions in other people. Another potential application is that we could have participants complete different kinds of memory tasks and see if different parts of memory are linked to different parts of the brain. So this is where we might be able to find some support for things like the working memory model, which you learned about in your memory topic. We can also study the brains of people with psychological disorders. So we might find that there are different levels of activity in different parts of the brain in patients with OCD, for example. So for them, we might find that the orbitofrontal cortex is more or less active than in a normal or healthy population. Okay, so quick evaluation. So it has very good spatial resolution, so remember that means it allows us to see which brain areas become active during a task with a good degree of specificity. And it's also non-invasive, so what that means, it doesn't require anything to be injected into you. It doesn't mean that, and you don't also need to use anything radioactive. So another kind of scanner that we don't need to know about is a PET scanner, which does the same sort of thing. It tells us which parts of the brain are active during tasks, but a PET scan requires that you have a radioactive isotope injected into your bloodstream. fMRI doesn't require that, so we can say that's a good thing. It's non-invasive. People generally don't like to have injections unless they need to. However, fMRI is relatively poor in terms of its temporal resolution. It takes about 1 to 4 seconds before the fMRI scanner can actually pick up that change in brain activity. They also don't show how different brain areas interact with one another. So it may be just as important to say that your frontal lobe is communicating with, say, your amygdala, as it is to say that those parts of the brain are active. So it doesn't show us the interactions between parts of the brain at all. And additionally, fMRI scans are very, very expensive. I think an MRI scanner is somewhere between like $10,000 and $15,000 or something like that. So very, very expensive to use. Right, so the next thing we need to look at is an electroencephalogram, and that's quite a complex word, so I'm going to break it down for you. So the encephalo part of that word is just from the word encephalon, which is literally just another word for the brain. It doesn't mean anything special, it just means brain. And the electro part just means that it uses electricity, so it uses electricity to measure the brain. And the gram part just comes from the fact that it produces a graph. So the EEG produces a graph showing the electrical activity on the surface of the brain, also known as the encephalon. Okay? It does this by measuring electrical activity across the scalp. So when you have an EEG done, as I did when I was at university, they'll place a big cap like this on, and it will be covered in electrodes, all of which will measure the electrical activity on the scalp directly below them. And it will then measure the changes in the electricity, which allows us to measure the electrical activity in the cerebral cortex, so only the surface of the brain directly underneath. So EEG is used to examine how activity in the cerebral cortex changes across time or between states. So it's quite generalized. You put it on and you'd keep it on for a good few hours, and we'd be able to see how brain activity changes between something like being awake and then the different sleep cycles. So you might wear one of those hats like this, an EEG machine, while you have a nap, and they'd be able to measure the way that your brain changes from being awake and then the different parts of the sleep cycle. So like the REM would be the deepest level of sleep. You might also use it to measure how someone's brain changes when they have a seizure. If someone is very prone to seizures, you could use that to see which parts of the brain are affected. And we can also compare neural activity while drunk and when sober. So you could put one on someone, give them some alcohol, and see how their brain activity changes when they become drunk. But the main thing to understand here is that you're looking for changes in states across time. It's very generalized. There's no specific thing being presented. You're just looking at changes in the state of being across a long period of time. So the EEG output would look something like this, and each one of these would correspond to one of the electrodes. Now, don't worry, even at university level, you would never be expected to be able to interpret one of these unless you specifically chose to do a study involving EEG. Even at master's level, like my friend did a master's in cognitive neuroscience, and she only had to do a tiny bit of this because it was directly relevant to what she was studying. So although it is a very complicated output, it's not something you would be expected to read even at degree level. So one advantage is that EEG is incredibly cheap compared to MRI scanners in particular. It's also non-invasive. You just have to wear something on your head, and because it measures general neural activity across time, it can tell us changes in the brain between different states. However, it has relatively poor spatial resolution because, as you look back, it can only tell us about activity between here, so that's not a very specific brain area in just this part here, compared to incredibly small neurons. The major flaw, however, is that it only measures activity on the surface of the brain. It can't measure those deep brain structures like the amygdala or any other parts of the limbic system like the hypothalamus, just the cerebral cortex. So the third one you need to learn about is event-related potential. So ERPs, basically they make use of an EEG machine, so it's the exact same procedure for how it's measured. The only difference is that rather than measuring general activity across different states of being, it measures activity in response to a very specific stimulus or an event. So, for example, you might have someone sitting at a computer screen and you might flash up threatening images. So, for example, a picture of a snake or a picture of a spider or something else which is scary to the participant. And you might see what changes in brain activity happen directly in response to that. Another potential application is that it might be able to prove or disprove free will. And this video is about the Libet experiments, which I'm going to put in the Google Classroom. I'm not going to show it now. And basically what he did is he showed that people's brain activity related to their behaviour before they themselves were consciously aware that they were going to carry out that behaviour. But like I said, the video will explain that in more detail. So evaluation for ERP is basically the same as EEG. It's cheap, it's non-invasive, it has excellent temporal resolution as well, however. So this can tell us about the changes in brain activity in response to that stimulus being presented to the millisecond. It's very, very good. However, same as EEG, poor spatial resolution, only measures activity on the surface of the brain, and it does require a great deal of control in this case. So even very minor movements or distractions can show up on the output. So when I had my EEG done, I kept kind of like grinding my teeth or just moving, and the person would shout through the wall to stop doing that because it would cause massive spikes on the EEG output, even though it was only a very small movement. So finally, post-mortem examinations, fairly self-explanatory. We can examine the brains of people who showed cognitive or behavioural abnormalities while they were alive. So the easiest one to give an example would be patient Tan. So patient Tan, if you remember from the last video that I did, he could only say the word Tan over and over again. So because we know that's very abnormal behaviour, once he had died, Broca examined his brain and found that he had a lesion or damage to what is now known as Broca's area. So we can look at people who show a wide range of different abnormalities, and then once they die we can see which parts of their brain are different to a healthy brain. So one of the positives about this is that it can allow us to examine deep brain areas. So whereas EEG could only show us the surface of the brain, this allows us to have incredible detail of all kinds of brain areas. So obviously we can take a particular brain area out and we can examine it under a microscope. We can kind of cut a brain up however we want once that person is gone. So very, very good spatial resolution. However, it's unable to establish causation. So it could be that the damage to that part of the brain is what caused their abnormal behaviour, but it could also be that an underlying medical condition caused a change in behaviour and caused the difference in the brain structure. It's also very difficult to replicate because we'd need to find somebody who's showing the exact same symptoms and wait for them to die before we could check it against the first brain we've examined. And also, I've said here, dead men give no consent. And even though you might think that, so some participants might be able to consent that you can study my brain after I've died, that's going to be quite difficult with people who might have very severe cognitive deficits. So for example, I can't remember the name of the case study now, but the man who only had who had no short-term memory, I think it was HM, he wouldn't have been able to give proper consent for what happens after he died because he wouldn't be able to actually remember giving that consent, and that might then have changed between one day and the next. Okay, so those are the 4 different kinds of neuroimaging that you need to know for your exam. Just to check that, I'd like you to see Google Classroom. I've uploaded a few questions. It should only take you about 10 or 15 minutes to complete those. I'd like those uploaded for Friday, please. Awesome, thank you guys.